The topic that we are going to discuss in this video is of prime importance in immunology. We know very well that the ultimate aim of an immune response is to eliminate or neutralize the threats to the body caused by an infectious agent or pathogen. Following infection, the first encounter between the pathogen and the infected individual is through the innate immune system that provides generalized immunity against all microbes, often resulting in an inflammatory response. However, there are more than one reasons that pathogens can avoid this innate immunity. One reason is that pathogens evolve ways so that they can't be recognized by the innate immune cells and then can't be killed or eliminated. So here's second type of immune system works that is known as specific immune system or adaptive immune system. It is a collection of specialized organs, tissues, and cells that can recognize the presence of the pathogen in much efficient way. Following recognition of pathogen, the cells of the specific immune system are stimulated to generate a specific immune response. Now this specific immunity involves both humoral or solubilized components and cellular components. Immunity generated by humoral or solubilized agents or mediators is termed as humoral immunity, while immunity generated by specific cells or cellular components is termed as cell-mediated immunity. The relative contributions of the two were widely debated at old time, but now it is obvious that both have very important role in body, and the full immune response requires the action of both. Here our concern is with humoral immunity that is also called as antibody-mediated immunity. Now, why is it called so? Well, the soluble components of humoral immunity are mainly composed of a fraction of serum that was called as gamma globulin or immunoglobulins. And the soluble active components in the immunoglobulin fraction of serum are now commonly referred to as antibodies. So we can say antibodies are the soluble components of body fluids that was previously called body humors that are responsible for the immunologic events under humoral immunity. That's why humoral immunity is also termed as antibody-mediated immunity. Let's learn more about what are antibodies. So, antibodies are actually the protective proteins that react specifically with the foreign agent, called as antigen, that has stimulated their production. These antibodies play a role in recognition of antigen and also activates other immune responses, like phagocytosis or complement, etc., to kill or neutralize these antigens so that they can't produce a disease or any harm inside the body. Antibodies make up approximately 20% of the plasma proteins. And the two key characteristics of antibodies are that they are usually very specific in terms of binding only on a particular antigen, and that they have a high affinity, meaning they bind antigen very strongly. After knowing what an antibody, let's look how it works. When any foreign agent that is alien to the body enters inside the body, it quickly gets recognized as foreign body because of its surface molecules that differs from that of those found inside the body. So after recognition, a number of protective mechanisms activates. Among them, an important one is antibody production. This production is done by specialized immune lymphocytes called B lymphocytes. These B cells recognize antigen by its specific receptors present on its surface called BCRs. This BCR is actually an antibody that is anchored on the surface of B cells. The antigen receptor does not recognize the entire antigen. Instead, it binds to only a portion of the antigen's surface, an area called the antigenic determinant or epitope. Binding between the receptor and epitope occurs only if their structures are complementary to each other. So once activated, it stimulates division of B cells into mature and identical clones of cells. Mature B cells are called as plasma cells, and these plasma cells then secrete millions of antibodies into the bloodstream and lymphatic system. These antibodies then binds to the foreign antigen, neutralize toxins or foreign bodies. provide aids in phagocytosis, that is, engulfment of pathogen, activates complement system so to produce proteins for killing invaders, provides long-term immunity as they persist for years after antigen encounter, and acts as first-line defense for mucosal surfaces as well. So this was general introduction about what is an antibody and how it works. This video will further boost your knowledge about the elucidation of antibody structure. That shows how experiments lead to determination of antibody structure. Then we will look into its further structural details. We will also learn about the classes of immunoglobulins. And lastly, we will learn about important functions of antibodies.